Welcome biologists. In this session we're going to be taking a look at the differences and similarities between the structure of prokaryotes and eukaryotic cells. Now eukaryotic cells we've already had a look and the organelles present there, but the main thing here is a eukaryotic cell has membrane-bound organelles inside, including a nucleus, whereas a prokaryote has no membrane-bound organelles and it has no distinct nucleus. So we're going to go through the structure of a prokaryote, and by the end of this, you should be able to then think back to what a eukaryotic cell is and then think about the differences between the two. So here I have a very simple diagram of a prokaryotic organism, and I'll go through each of the parts of the prokaryotic organism. So first of all, we have a capsule. It's not always present. So in an exam, I would always go with the cell wall, it being on the outside, but sometimes they can have capsules present, which are involved with protection and strength. Anything in a red box is taken from the mark scheme, and this is what you need to know. So cell walls in a prokaryote, these are made from either murin or peptidoglycan, and yes, you need to know those names, and they provide a high tensile strength for the organism and therefore protection. Next one is the cell surface or the plasma membrane. This is the only membrane you'll find within a prokaryote, and it's involved with allowing substances in and out of that cell. Next one is the cytoplasm, which is the site of chemical reactions. We also have ribosomes, which are involved in protein synthesis. Now, the main difference here between a prokaryote and a eukaryotic cell's ribosomes is the size. In a prokaryote, the size of a ribosome is 70S, whereas in a eukaryotic organism, they are 80S. In a prokaryote, the ribosomes are slightly smaller in size, but they still do the role and still do the job of making proteins. In a prokaryote, we have plasmids, which are circular pieces of DNA found within the cytoplasm. And these circular pieces of DNA, these can be passed on to other bacteria and to exchange genetic information, such as antibiotic resistance. So if this particular cell here on this picture had the genetic information which coded for antibiotic resistance, this particular organism could pass that information through a plasmid, through the pili to other surrounding bacteria. And again, not all bacteria have the pili on either. Another feature we have here is a nucleoid. And this is like the bulk of DNA within inside a prokaryote. Again, not all of them have this, but the key thing here is that this is circular DNA found within a prokaryote, whereas in a eukaryotic organism, it's linear DNA. We also have the flagella, which is not present in a eukaryotic organism, and in a prokaryote, in some prokaryotes that have these are involved with propelling the cell in a certain direction, and they're involved with movement of that cell. And yes, they do require ATP. Now, ATP can actually be produced from something called a mesosome. A mesosome, again, it's not in all prokaryotes, but a mesosome is the folded regions of that cell surface or plasma membrane. And that is the process by which ATP is made on those folded regions of that cell surface or plasma membrane. Here is a picture of a mesosome that you can see here on the left-hand side, that folded membrane by which the prokaryote can make ATP. Here's a typical exam question when it comes to prokaryotes and eukaryotes and different cell components. Now, before you take a look at something like this, it's really important that you know that chloroplasts and mitochondria have evolved from prokaryotes. So therefore, the internal structure of a chloroplast and a mitochondria will be similar to that of a prokaryote. And what we mean by that is that the DNA found within a chloroplast and a mitochondria, because they do have small bits of DNA, there'll be circular DNA found within the chloroplast and the mitochondria, and they'll also have smaller ribosomes present within the chloroplast and the mitochondria. They'll have those 70S ribosomes, not the 80S ribosomes, which are found within the cytoplasm of a eukaryote or attached to the ruffia of a eukaryotic organism. So for this particular question here, you need to think about it in terms of this Venn diagram, and it might be worth practicing writing out below so that you know where everything goes. But this is a very typical exam question that can be found on the OCR paper. So if you want to pause it, have a go at doing that question. Might be a good idea, but the answer for this is D, which is the bottom one. And that's it, guys, for this one. Good luck with your